So hello friends, today we will discuss the mechanism of beta-lactam antibiotics. Okay, so mainly these beta-lactam antibiotics inhibit the cell wall synthesis. Okay, so how it will inhibit the cell wall synthesis we will see through the diagram. Okay, so if you see here, this is the bacterial cell. Okay, so if you see the bacterial cell, this is the cell wall. Okay, what you will say, this is the cell wall. The inner membrane is called cell membrane. Okay, and this in between two means a cell membrane and cell wall, this space is called periplasm. Okay, actually if you see, this cell wall is very thick in gram positive bacteria, while it is thinner in the gram negative bacteria. Okay, and if you see the chemical nature, in between this one, this cross link structure is present. Okay, and it is much found in the gram positive as compared to gram negative bacteria. Okay, so if you see, in this is the cytoplasm. Okay, inner space is filled through cytoplasm. Okay, so if you see, I have cut the one part of cell wall. Okay, so if you see, this is the, I have cut this part, means this is the cell wall part. So if you see the inner chemical composition, then you will see that it is the structure of like this. Okay, so these structure are the nag and name. Okay, and if you see in this this is the name, this is the name, and this is the NAG. Okay, it is called N-acetyl muramic acid, while it is called N-acetyl glucosamine. Okay, and this name containing 5 outgrowths, and this outgrowth is called amino acids. Okay, and several types of amino acids present, and the terminal amino acid is called alanine. What is called alanine? Okay, and this amino acid is, number of amino acid is 5. Okay, so if you see how this layer will be formed, if you see here, so you should be consider the chemical nature of N-acetyl glucosamine and N-acetyl muramic acid. So if you see here, I have mentioned this, since N-acetyl, if you see the name of this one, this is the N-acetyl glucosamine. Okay, so glucosamine is the sugar. Okay, N-acetyl means N-acetyl, what is this, is N-acetyl. Okay, means this N-acetyl moiety is attached to glucosamine. Okay, so if you see, this is the structure of 5 carbon sugar. Okay, this is the CH2H, each carbon is sugar having primary alcohol. Okay, this is the H and OH. Okay, and before you should be see, you should watch the structure of carbohydrate, so you will definitely you will see the very clear way the diagram of carbohydrate or glucose. Okay, so it is the structure of glucose, and this terminal OH, and this is the OH. Okay, so very simple. If you want to see the to draw the structure, then what happen here, this is the chain of carbon and 1 oxygen. Okay, means 5 carbon is present in the ring. Okay, so I have mentioned the 5 carbon, and if you see each glucose having CH2H. Okay, remaining is OH and 1 OH and 1 H. Okay, so you will draw the structure of this H and OH. Okay, and this carbon is containing NH, N-acetyl moiety, so you will attach NH and COCH3. Okay, and like that OH and H will be present. Okay, so this is the N-NAG. Okay, the name of this is NAG, and this name is N-acetyl muramic acid. In this muramic acid, okay, so acid part is attached through this one. Okay, and if you see the cis, and the similar fragment is like COCH3. Okay, so means what is extra moiety is here, COCH3 and COH. Okay, so this is the difference between N-acetyl glucosamine and N-acetyl muramic acid. Okay, so if you see here, when these join together, the one water molecule will be released out. Okay, means dehydration step takes place. Okay, so one OH will be released from muramic acid and one hydrogen from hydroxyl group will be replaced from NAG. Okay, so it is the formation of NAG and NAME complex. Okay, it is also called N-acetyl glucosamine, N-acetyl muramic acid, pentapeptides. Okay, because 5 amino acid joined to peptide bond. Okay, so it is the formation of NAG and NAME. Okay, so if you see this one, okay, so it is the NAG and NAME joined together through which bond? Okay, so glycosidic bond. Okay, so if you see here, this is the complex, this is the NAG NAME joined together through this chemical nature. Okay, and if you see in this 5 amino acid is present here, so I have attached 5 amino acids, and through when it joined together it provides the rigidity to the cell wall, which portion it will provide the rigidity cell wall. Okay, so if you see two enzymes is here, one is transpeptidase and another is DLA carboxypeptidase. Okay, so transpeptidase, the function of transpeptidase is that it replaces one terminal amino acids. Okay, and DLA carboxypeptidase joined means through fabricate both these amino acids of muramic acid and another amino means this join together through this one. Okay, so the function of join together, this takes place means D-alanine carboxypeptidase join two terminal amino acids of two NAG NAME. Okay, which is anti to each other. Okay, so the function of transpeptidase is replacing the one terminal amino acids. But what happened here, if you see the structure of this one, okay, so this is the transpeptidase. Okay, but what happened if penicillin is present, if any patient suffering from bacterial diseases, and when it takes the penicillin or amoxicillin, there are several types of beta lactam antibiotics. Okay, so the main framework, the basic moiety, you can say this is the one 4-membered and one 5-membered. Okay, so this framework is present in the penicillin. Okay, so it what happen here, this penicillin attaches to the terminal amino acids. Okay, and when it comes means when transpeptidase comes, it means it dismisses means it cannot join on that places because penicillin is already present. Okay, so by which the functioning of cell wall synthesis will be breakdown. Because why? Because if penicillin will be reaches here, the function of transpeptide does not takes place. Okay, by which these two amino acid will does not attach. Okay, so that's why the process of cell wall synthesis will be inhibited here. Okay, so here functioning of transpeptidase becomes inhibited, so that's why it does not means amino acid does not attaches to each other, and by which rigidity becomes less. Okay, rigidity and cell synthesis will be blocked here. Okay, so by which osmotic pressure of inside the cell become much more, and by which it becomes bursts. Okay, so since it breaks the cell wall, so it is also called bactericidal disease, bactericidal drug. Okay, so it is the basic moiety of it is the basic mechanism of beta lactam antibiotics. Okay, so it is all about. Okay, so if you see if you see the uh, resistance of some bacteria, so in the what happen in that cases we will describe in the next lecture. Okay, so thank you for this.